G-value paradox. This one's going to be a little shorter than most. Just one kind of major key concept and then two explanations for why. Before we get to the G-value paradox, though, I'd like to review and extend a little bit our discussion about why genomes are so big. So remember, this came as a big surprise because as we began to annotate this genomes that we had sequenced, we found that particularly in the eukaryotic genomes, there's just tons and tons of non-coding DNA. That didn't apply so much to the prokaryote genomes, and we'll look at that in Unit 3.1D. But this was a surprise, and so people were trying to figure out why. Why do we have so much DNA? And there were some ideas, again, about the structural effects on cells. There's a correlation there, but that doesn't seem to be a good explanation. It's not really causative, they're just correlation. We don't know what causes it, if one is causing the other, and that's not really a good explanation. And so we're just basically left with this idea that there are mutations that occur. Some of those mutations enlarge the genome, and some of them make the genome smaller. So you should be familiar, maybe not in great detail, but with all of these mutations. And this is in relation to prokaryotes, but all of these things also apply to eukaryotes. So we could add the eukaryote label to this. So if we get an extra gene or an extra part of a chromosome or a gene duplicates, if we lose a portion of a gene, that makes our genome smaller. And so if there is a bias one way or the other, then over many, many generations, we will either add genome size or subtract genome size. Now in eukaryotes, there are two additional major, very, very important factors that add DNA to the genome. So let's list those and we'll talk about their impact in Unit 3.1E. So eukaryotes, and if we were looking at, say, the salamander genomes versus the frog or the chicken or the fish genomes, we would probably see that these are the two major factors that are making these salamander genomes so massive. And they don't really provide any inherent advantage. Well, they don't have an advantage if you have more of it. It's important that these things are there in eukaryotes, and they play a major role in eukaryote genome evolution, but having a lot versus a little is not really a big difference. But some organisms just have this happening more, and so their genomes get very big. The first thing is a transposable element, and there are several types of transposable elements, but the ones that we're most interested in are called conservative transposable elements. And there are many different subcategories, but basically that means a piece of the genome that codes for its own kind of a cut and paste mechanism. A copy and paste mechanism. So they make copies of themselves and then reinsert themselves into the genome. Sometimes these are called selfish genes because they don't really provide any advantage to the organism, but because they make extra copies of themselves, they get lots of them in the genome, they make the genome larger, and this is a major important factor amongst the eukaryotes. And then, of course, introns. Prokaryotes, by and large, don't have introns. There are a few minor ones and a few small groups of bacteria, but they don't play a major role. So introns are the non-coding DNA that is in the middle of genes, and it's kind of a very weird way to organize a gene. We think of a gene as a very specific set of instructions to make a product, usually a protein. And that's true, they do that. But imagine looking at an instruction manual for some sort of an important, let's say you wanted to learn how to build a remote control car or something, and you open up an instruction on how to build one part of that car, and you see the first few sentences are really great, they help you, and then suddenly there's random letters, and you have to skip ahead a couple pages, and then that's the rest of the instruction. That would be like introns. Introns are like that random, non-useful information in the middle of a gene. And so we'll talk about why they're there in a later unit, but just think about that. And so they add to the overall non-coding DNA of a eukaryote. They make some eukaryotic genomes very, very large. So all of this can be put under the general category of non-coding DNA. And that's the simple explanation for the C-value paradox. Where the non-coding DNA comes from is a little bit more complicated. But the C-value paradox is simply, we've got mutations that occur. They bring DNA in. If there are more of those than remove DNA, genomes are going to get big. And organisms with really big genomes just have more of those factors that bring in DNA, and it may not even provide any sort of advantage. They just have it, and if it's slow buildup over time, there's not really a disadvantage for each of the individual mutations that brings in more DNA, and so they end up carrying more DNA around. Sometimes this non-coding DNA is even called junk DNA. That's really a misnomer because it doesn't have zero function. It's not totally useless, but its overall impact on the phenotype is much more diffuse. It's spread out across a huge amount of DNA and only has a small overall impact compared to the genes themselves, the coding regions. Okay, so finally on to G-value paradox. The G-value paradox came about as scientists began to examine, map, and annotate genomes. And they said, okay, we know now that there's lots of non-coding DNA, and that explains why genomes are so big. So let's ignore the non-coding DNA. Let's look only at the genes, and maybe even only at the protein coding genes. So realize there is a slight discrepancy. So coding genes are genes that code for anything, including RNAs. You could even throw in short microRNAs into that category. Pseudogenes that maybe are transcribed but not translated. There's a number of ones, and we're just beginning to understand some of the diversity of the non-coding genes. But really they make up a small minority. So blue includes everything that is transcribed. And then the orange here is only the protein coding genes. So notice it's 90% in some species, 75, 80% in other species, but protein coding genes make up the diversity. And so scientists decided to look at only these coding sequences. And surely they thought, okay, more complicated organisms, like mammals, for instance, would have more tissue types, they're more intelligent, they have these complex interactions. By most definitions, mammals are more complex than, say, a clam, right? Or even a bird, which are fairly complex, but mammals are kind of in the highest class. And so they thought, if we look at the genes of mammals, we're going to see way more genes, right? You need more instructions to build more complicated organisms. And although it's not as strong as the C-value paradox, there still is a paradox here. So G-value, the number of genes you have, is not one-to-one predictive of an organism's complexity. And you can see it here. So let's just use an example. The pufferfish, which is way simpler than you are, has more coding genes and about the same number of protein coding genes as you do as a human. So even though it's simpler than you, it has more by some counts and about the same amount by other counts of these coding sequences. So that's a paradox, right? We would expect a fairly simple fish to have fewer genes. Now, although in concept, this is just about as simple as the C-value paradox, right? Don't get them confused. So review. C-value paradox, genome is not predictive of complexity and vice versa. G-value paradox, the number of genes you have is also not predictive of complexity or vice versa. So the explanation for the G-value paradox is a little bit more complicated, and you need to know these three things. So number one, your complexity is not only predicted by how many genes you have, it's how those genes are connected together. So in humans, the same number of protein coding sequences, are roughly the same, are connected together in way more complex genetic networks. So in a way you could say, hey, if you gave somebody the same toolkit, but then they could take two of those tools and connect them together and make a third tool where somebody else didn't know how to do that, with the same number of tools, you could have a more complex outcome. Okay, so we have these more complex regulatory networks. In addition, remember, the non-coding DNA is not completely useless. It does play a role. And so by only counting the genes, we are discounting a portion, and it might be a minority, maybe 20, 40% of the overall complexity of organisms, but it's still an important contributing factor to the complexity of these organisms. So we've got regulatory genes. We may have other, like microRNAs that aren't being counted, particularly in the protein coding sequences, that do play a role in the complexity and the phenotype of the organism. So remember, the non-coding DNA is not completely useless, and so it does play a role. And then finally, by just counting genes, we miss part of the picture of the overall outcome of transcription and translation, because after a gene is transcribed, it can be differentially spliced, for instance, or there can be different isoforms, or there might be massive allelic variation. So just one gene does not automatically come out in one protein product. And I'm going to give you one example here. You can look it up and read up on it if you want. This is maybe the best example overall and explains a portion of mammalian diversity. And these are immunity genes. So mammalians have a much more complex and adaptive, you could say, and even proactive immune system. You don't really see that in other organisms. And so we have, for instance, a single antibody gene that is transcribed, but then it's differentially spliced in all these hundreds upon hundreds of thousands of combinations that make many, many, really millions and millions of different combinations of antibodies. And these antibodies are one of the major things that helps us recognize foreign invaders. And so that makes mammals more complex, despite not having as many genes, or maybe just as many genes, as some simpler organisms. Okay, so remember those, review those, go over those three explanations for the G-value paradox, and that wraps up this discussion.